Hi everyone, I am Anita and I'm a PhD student at the Institute of Plant Breeding, Genetics and Genomics here at the University of Georgia. In this video, you will learn how to isolate protoplasts from poplar leaf tissue. Let's dive right in. First of all, materials. We need freshly harvested poplar leaves, a razor blade, a spatula, a 40 micron and a 70 micron strainer, an electric pipette together with a 25 milliliter serological pipette, and a set of micropipettes to prepare the enzyme and storage solution. I have already prepared 20 milliliters of each using the recipe shown on the screen. These two solutions are exactly the same, except that the storage solution does not contain any of the cell wall digesting enzymes shown in red. For more information on how to prepare these solutions, take a look at the reference at the end of the video and in the video description. We are also going to need a couple of extra tubes. We're going to start by cutting our leaves into really fine, about 1 millimeter wide strips. The purpose of this process is to expose as many cells as possible to the action of the cell wall digesting enzymes. This might take a few minutes, but the result should look just like this. At this point, we can transfer our leaf strips into the tube with the enzyme solution using the spatula. We want to make sure that the leaf strips are completely covered by the enzyme solution, and so we can gently push them down with the spatula. After making a hole on the tube cap, we can proceed to vacuum infiltrate the tissue at 600 millibars for 20 minutes, with a stop at the 10 minutes mark. After vacuum infiltration, we can transfer both the enzyme solution and the leaf tissue onto a new petri dish that we labeled accordingly. We can help ourselves with a spatula, and we need to make sure that the tissue is evenly distributed over the petri dish surface. Once we've done that, we can close the petri dish, grab some paraffin, and seal the petri dish before transferring over to the shaker. We're going to leave our petri dish on the shaker for about an hour, 45 minutes at 50 RPM, with an additional 5 minutes at 80 RPM to facilitate the detachment of protoplasts into the enzyme solution. Now we can harvest our protoplast. To do that, we need to use our stripette to collect the enzyme solution and then filter it through the 70 micron filter and the 40 micron filter in this order. After filtering, we can spin the filtrate at 100 G for 5 minutes at 4 Celsius to pellet the protoplasts. At this point, we can discard the supernatant and resuspend the pellet with the protoplast in some storage solution. Alright, now that we have our protoplast, we can look at it at the scope. And this is how it looks like at the microscope. We see that the protoplasts look like nicely rounded cells, as indicated here by the black arrowheads. Now we see that there's also some debris left over in this preparation, which might be an issue depending on the end use of your protoplasts. So depending on the degree of purity that is needed, you might want to consider additional purification steps such as flow sorting, for example. Well, that was all I had for you. This was leaf protoplast preparation for poplar. I hope this was helpful, and thank you for watching.